Good afternoon, everyone. We're just going to wait for a few seconds to allow everyone to join. Okay. So, welcome to this webinar, and thanks very much for joining. Today's webinar is Optimizing Cell Counting in Biopharmaceutical Manufacturing. I'm your host today. I'm Annemieke Karyotis from ChemoMetec. I'm a field application scientist. ChemoMetec, just quickly, is a global manufacturer of cell counting equipment and cell analysis equipment. It was founded in 1997. We now have customers in 112 countries and about 150 full-time employees. NIBRT and ChemoMetec have been collaborating since 2020 on training activities and research activities and how to optimize cell counting and manufacturing processes. Therefore, gives me great pleasure to be able to facilitate this webinar today. And our speakers are from NIBRT, of course. It's Bergin and Pritchard. Just a couple of housekeeping notes. Presentations will probably be about 20 minutes, and then we're going to have a live Q&A session lasting about 10 minutes. You can ask questions using the Q&A functionality at the bottom of the webinar. I'm pretty sure we've already got actually a lot of questions beforehand that people have emailed in, so we probably won't be able to get through them, but rest assured they'll be saved in the Q&A function, and then we can afterwards email you and get in touch with you to answer your questions. And if there's any technical issues, you can use the chat function to let us know if the sound or the picture is not working. So I'll hand over now to Adam.
Thank you very much. So thank you for that introduction, Annemieke. Very happy to be here. Thank you everybody for attending. So as was mentioned earlier on, my name is Adam Pritchard, and I'm a training team lead in NIBRT. So for anyone who isn't aware, NIBRT is the National Institute for Bioprocessing Research and Training. And based on the length of that name, you can understand why we shorten it to NIBRT. And the aim of this company really is just to support the biopharmaceutical manufacturing industry, both in Ireland and abroad. So we were actually set up by the IDA, who are the Industry Development Agency in Ireland. And the idea was that if there was a bespoke training and research institute based on the island, it would support biopharma manufacturing. And thankfully, this has been quite a successful endeavor. And as you can see there, in 2020, the biopharmaceutical sector in this country actually accounted for about 93 billion euro in exports, it's a huge proportion of the Irish export industry because we are a very small country really in the grand scheme of things. Now, everything I've mentioned so far really is based around Ireland, and that is a major focus of ours, but we do have a worldwide trainee portfolio. Based on this map, you can see that pretty much no continent is untouched by NIBRT, bar Antarctica, obviously. And we have had trainees on site and for remote training activities from countries across the globe. Now, because the facility is of finite size and we have a finite number of trainers, we have begun to offer our training curricula through our Global Partners program. So if you want to get training in the NIBRT way, you can now do this in a range of different countries. So in the United States, in Canada, in Korea, China, and in Australia, there are institutes that are delivering NIBRT training, essentially. And one of the things that's been at the core of NIBRT training for as long as I've worked here, and prior to that, has been the upstream cell culture process as part of the manufacture of recombinant protein therapeutics. So anyone who has any kind of background knowledge in this area will understand how integral to the manufacture of these products cell culture is. And one of the key operations that is conducted in the vast majority of these facilities is cell counting. So when we have trainees on site and even through remote training platforms, we try to take them on a journey through these concepts. So we begin by describing the purpose of cell culture in and of itself as a protein production platform, how cells are subcultured then in order to maximize growth, and how different pieces of equipment may be utilized in order to achieve that for both adherent and suspension cells. And intrinsic to all of this is the application of cell counting. So for people who work in manufacturing environments, some of whom don't have a particularly strong biology background, it's very important to build up this information over time and to lay the groundwork, lay the foundations, and then add detail as you go. So from a manufacturing context, we're introducing this concept of monitoring growth, checking growth patterns are appropriate, and that is the purpose of cell counting in that context. In a PD or a process development context, we're talking about developing subculturing strategies, feeding strategies, and the requirement for cell counting in that. Now, as I mentioned, we're trying to take trainees on a journey, and we do this through our practical training as well. So we'll often begin a course or a practical session with a demonstration and some training on manual cell counting. So using trypan blue exclusion, using hemocytometers, and again, working on the fundamental concepts behind these activities. Now, again, anyone who has experience in the industry is probably well aware that manual cell counting isn't necessarily the done thing. I've used the term relic of the past here, which is probably a little bit harsh, but with that said, it's not something that is often done, primarily due to the subjectivity that is inherent in it. So if you're working in a GMP facility where reproducibility is of paramount importance, and we want to minimize variability between operators, we don't want to have a manual operation really. With that said, microscopes are still a very important part of the in-process checks that companies conduct, more so to do with contamination assessment. And it's often both the earliest and sometimes the only opportunity for an individual to detect contamination in a cell culture sample. So it is a little bit of a lost skill, but certainly from a cell counting point of view, it's not necessarily the done thing anymore. So what I would say is that of the trainees that NIBRT has on site and has had over the last 11 years or so that we've been in operation, the majority would use automated cell counters. And the trypan blue method with these automated cell counters has to some extent become almost like an industry standard. You have the major benefits of reproducibility, as I'm sure you're aware, and consistency, and the fact that every time it's conducted, the operators have a set series of steps they perform that are quite reproducible. But that's not to say it's the optimal way of achieving it. And while we do deliver a lot of training in this area, and we talk about the methodologies and some of the pinch points and some of the technical issues that can happen with fluidics, etc., and maybe how to troubleshoot them, there's always room potentially for improvement and scope for change. Now, historically, biologics manufacture has been risk averse. It's a regulated industry. People don't like changing things. But I think the industry as a whole is somewhat in flux at the moment. And we're certainly seeing much more openness in terms of adopting new techniques that have the same benefits in terms of reproducibility, ease of use, reliability, certainly of the equipment is vitally important. But if there is an option that provides more flexibility and maybe provides information that wasn't previously available, we do see more and more companies interested in alternative techniques. So things like nucleocounters are becoming increasingly interesting to companies. And as a result, we adapt our training to showcase that. So not only do we try to train on the past techniques, on the present techniques, but we also try and adopt what we think are going to be the future techniques as well. And we try and upskill trainees in advance, meaning that if the equipment ends up on their site, it's not something that they're shocked by or is completely new. So that's it for me in terms of my kind of description of the NIBRT training outlook on cell counting, past, present, and future. I appreciate there may be some questions. We're going to address them towards the end of the presentation. Now I'd like to pass you over to my colleague, Adam Bergin, who's going to discuss a bit more about a case study that he has performed in this area. And thank you very much.
Thank you very much, Adam. So like Adam alluded to, I'm going to talk to you a bit more about some practical hands-on experience that I had using the ChemoMetec system. And just to confuse you that there is another Adam, so I appreciate that. I'm on the research side of the work that's been conducted in NIBRT, and I've just recently submitted my thesis for my PhD studies. So hopefully soon to be doctor after my name now, after this. So just to bring you onto a bit of the background that Adam had started talking to you about, and that is the use of trypan blue in cell counting applications. It's important to know that trypan blue is large in its molecular weight. And what happens is that it is excluded from cells that have an intact membrane. When cells lose their membrane integrity, that will allow the trypan blue dye to enter the cell and stain the cytoplasm. And when we look under the microscope at a population of cells, we will see cells that have been stained with trypan blue, those that haven't, and then we ultimately count those populations and express that as a viability of the population as a whole. Now, the use of trypan blue is quite outdated. That is, you can look back 100 years to when trypan blue was first cited in its use as a cell counting method. So that'll tell you how long it's been in use in industry. But one of the big issues that we have with the use of trypan blue is that it is now considered a carcinogen by the European Union. And not only that, it's potential mutagen, and it can be toxic to the reproductive system. So that's why we want to try and minimize the use of trypan blue as much as possible as a cell counting application. Now that leads us to the question, what other methods are available? And I'm going to talk about the use of the NC202 system as a potential replacement for trypan blue counting. But first of all, to give you an understanding of how we use trypan blue, so it's typically used in a 0.4% solution that's added in equal volumes to a cell sample. We incubate that cell sample for 2 minutes, and then we'll pipette our sample onto the likes of a counting slide or a glass slide with a cover slip. And then we can count the cells using 2 different methods, either manually with a hemocytometer, or we have automated counters which can now detect the cells automatically and express the figures. So that's what's routinely used in industry, and it's what we would use in our labs also. And again, it's just getting across that point is that we see that cells that haven't been stained are considered live, and those that have been stained are dead. So that gives us our indicator of whether the cells have died or not. Now, what's interesting, and we'll talk about this as well, is that there could be other indicators of cell death that using some of these counting systems that we may be able to look at in the future, depending on the system we're using. And I'll talk about that later in my presentation. So for the use of the NC202 system, what we have is we have little cassettes, and within those cassettes, we have a couple of dyes, fluorescent dyes that are. So we have acridine orange and we have DAPI. So in the case of acridine orange, we will see that it will fluoresce green under a fluorescent microscope, and acridine orange is permeable to cell membranes. So we will get staining of all cells with our acridine orange. However, DAPI will fluoresce blue, and that will only fluoresce when it has bound to nuclear DNA within the cell, but it's impermeable to intact cell membranes. So the cell will have to have died in order to uptake the DAPI stain and see that blue fluorescence. So that's how we can then go about calculating our populations, is that we measure cells that either show just green fluorescence, or we see cells that have green or blue fluorescence, and then that fluorescence is then detected by the NC202 system. So how we went about using this in our lab was quite simple. We had benchtop bioreactors and we grew cells, a well-known cell line, a CHO-EG2 cell line in BioGrow CHO media. We'd inoculate these bioreactors at 3 by 10 to the 5 cells per mil, and we ran them in a batch culture, which we sampled daily. So we would take counts in triplicate with our trypan blue counter, but we would also take counts with the NC202 cassettes that were provided. And one advantage straight away from using the NC202 is that the assays are much quicker, we found. That is, it took less than 30 seconds for us to load the cassette and then run it on the NC202 counter and get data back from the system. So that was one immediate advantage where you're not having to pipette on samples and then wait 2 to 3 minutes in order to get data from our trypan blue counter. Now, to have a look at some of the data, what we see is we see very good correlation between the NC202 data and trypan blue counts during the first 4 days of the culture. After that point, we started to see a loss in viability in our culture. And that's where we see a bit of a difference in terms of the counts. However, one interesting aspect, and can be explained for why we see some of these differences, is that we have what's called a debris index on the NC202. Now, cell debris in itself isn't a well-defined entity when it comes to cell counts, but what we know about debris is that it forms when we get cells entering pathways like necrosis or apoptosis. That is, the cell disintegrates and we start getting the formation of small bits of debris and particles in our culture. So what will happen with a loss of cell counters is that that debris will get counted as either total cells in the population, and you might get a spiking or an increase of the number of cells being counted towards the later stages of the culture. So that's where this debris index can be used as an indicator of potential cell death in the processes. And for us, it was something that was very interesting to monitor as part of our work in the lab. So just to conclude on what I just talked about here, trypan blue is commonly used in counting applications. However, in our opinion, we're seeing this as becoming quite an outdated technique when it comes to monitoring cell cultures. More modern fluorescence counters like the likes of the NC202 are shown to be quick, reproducible, and ultimately, particularly as somebody who works in a lab, it minimizes our contact with harmful reagents such as trypan blue. So that's always something that's greatly appreciated in lab settings. And also to restate what Adam was talking about in terms of reproducibility of the systems, by using the NC202, we could eliminate a lot of errors that we see in labs where we could have different personnel counting cells, or we could have different techniques when it comes to staining the cells and pipetting. A lot of those differences and errors can be eliminated using the NC202 cassette. So that's something that's great in terms of reproducibility of the data that we're seeing also. What we saw was that the data was very consistent with trypan blue counts during the stages of the culture where the viability was high. However, we saw slight differences at the later stages of the cultures, and that can be, like any counter, explained through different methods or different ideas like the formation of cell debris later in the culture, which may influence that data going forward. So that's concluding my talk and just some of the experiences that I've had with the NC202 system. And like we were saying, if you have any questions for us after, we'd be glad to take them. And at that, I'm going to pass over to Annemieke, and then I think she can talk to us through some of the questions that you may have. And thank you very much for listening.
Thank you very much, both Adams. They were really interesting presentations. I was wondering if I could trouble the audience for a very brief poll. And also if you could if you've got any more remaining questions, you can put them into the Q&A just now. So I'll just launch the poll. We just would like to know a little bit about you, the audience. So the first question is, how do you count your cells? And the second question is, do you have a training need at your site? And the third and last question is, what stage of product development are you at? And I will share the results of the poll in a minute. I can see everybody's clicking away here. Right, so I'll just give it another few seconds because I can still see lots of numbers coming in. from Paul. Right, so as I said before, we got an awful lot of questions emailed to us actually, with people registering. So we'll just go through, I just clumped them all together as to what were the most common questions that we received. There was one here, have you come across any study regarding impact of anti-foaming agents on cell growth?
Yeah, I could start off with the answer there on that. There's definitely studies that have been conducted using anti-foam in cell culture processes. Just to give a bit of background in terms of why anti-foam is added, it's quite often to ensure that enough oxygen is being given to cells that you would have an element of foaming that is true sparging of gases into bioreactors or other vessels. And ultimately, you want to try and minimize the amount of anti-foam additions throughout your culture. And there's a number of reasons, and I think Adam would be best placed to give you a bit more information on that. But like I said, ultimately, because it can impact viability because you're adding in components that ultimately don't want to be adding to the culture because they're additional, you might see some effects on viability long-term if the concentration of anti-foam in the culture became too high. Would you have anything to add to that, Adam?
Yeah, absolutely. And what we would see more so anecdotally from the companies that we would deal with is that immediately upon addition of anti-foam, they have increased controller output for their oxygen control, because it's a known issue, I suppose, with many types of anti-foam agent that there's a detrimental impact on oxygen transfer. So basically oxygen is less available to the cells immediately afterwards. So oxygen usage is staying stable, but oxygen availability or transfer decreases. So the controllers need to increase their output, so more oxygen flowing in, higher agitation rate, etc. And any kind of fluctuations in that regard really are something you want to try and avoid. So much like you've described from an industrial point of view, even at 10, 20,000 liter scale, you're trying to minimize the anti-foam addition. With that said, foaming is an issue, particularly with single-use bioreactors actually that are operating at kind of lower pressures. So it is kind of a necessary evil, for want of a better expression. But yeah, there are documented studies that we can point you towards that kind of outline some of these concepts.
Okay. And then for my part, there were an awful lot of questions regarding how do you count aggregated cells? So ChemoMetec has a protocol or a technology, so where we can count aggregated cells, and what we do is we use a lysis buffer. So you take your aggregated sample, you add a lysis buffer, and what that does is ruptures open the cell membranes, releases nuclei, and then once the nuclei are released from the cells, you can count total nuclei in lieu of total cells per mil. So that's basically a method of how we do it. But we will, in any case, for all of those who did ask about aggregation, we will be in touch via email and give you a more in-depth explanation of how the assay works. Okay, right. So, and then there was also about GMP cell counting methods, it says here in the box.
Yeah, I can feel that one. I suppose it depends what you mean by GMP. I mean, if you're looking at something that's, 21 CFR compliant, etc. Yes, absolutely. And as I said, of the companies we deal with, the majority use things like Vicell, CDEX for things like MAB production. They work perfectly well, but there's no reason why you can't really validate any cell counter you want in those applications. And we also deal with companies who work in the stem cell manufacturing space, and they have different requirements, and they may have a different cell counter like the Luna. Or then you also have things like the NC202, obviously, which we widely see companies we train with who work maybe in the cell therapy space, things like CAR-T. But if you're talking about GMP in terms of data integrity, then there are quite a few available. But as I mentioned, the industry tends to be quite averse to change, so tend to proceed with what they know. And Trypan Blue has kind of filled that niche. But as I also said, we're seeing more and more companies who are interested in exploring different ways of doing it. And things like nuclear staining, just, they offer an alternative, to what is established, but that is exciting to a lot of people, and they're very keen to try something new and see how it can give them more data maybe than they were able to achieve previously.
Yeah, okay. And then this is sort of slightly related because it says, what do you believe is and should be the industry standard automated counting method? It's a bit of a tricky one.
Yeah.
Go on, Adam.
I was just going to say, look, when it comes to gold standards of counting methods, there's a lot of different technologies available, and it changes in terms of the amount of cells you're counting at any one time. But what's most important is, I think you have to be able to have a system that's calibratable, and either you run your samples off a standard curve and ensure that the samples, when you do test them, are reproducible over time. And that's something that's very important, because ultimately that's what you want from a counter, is that the data that you're generating is reproducible. That to me is what would make it a gold standard counter, is something that you can rely on to give you accurate counts time and time again. So just from a lab point of view, that's, in my opinion, what I would be looking for in a cell counter in that scenario. Yes.
I mean, we regularly run linearity tests, making sure that, if the count should be linear and, within certain CV, so there shouldn't be too much variation if you count the same sample 3 times, all of these standard tests, really. So I think we're almost coming now to the end of the Q&A session. So what I'm going to do is share the results of the poll. Let's see, can you all see that? Let's see, so yeah, so we got 48% nuclear counter, 27% hemocytometer, and then another one on the 24%. And then, do you have training needs at your site? So we've got some upstream all the way across, really. And then here, oh, that's really interesting. What feasibility, preclinical, it's pretty much an even a quarter each. Well, thank you very much for sharing that. That's really interesting to know. Okay, right, well, I think that's it for today. But if you have asked questions, we will definitely be in touch, and you've put them in the Q&A session. Okay.
Thank you, everybody.
Thank you, everybody.
Thank you very much, everybody.